early replication of developmental domains in mouse embryonic stem cells, in which we aimed to better understand the role of master transcription factors in replication timing. Please note that the online presentation is redacted from the in-person poster. Replication timing refers to the temporal order in which the genome is replicating during S phase. It is a cell type specific process that is crucial for the maintenance of the epigenome after replication. Replication timing is highly correlated with gene expression, histone marks, AB compartmentalization, and other genomic features. Early replicating regions correlate with A compartments, active histone marks like H2K27 acetylation and transcription. Conversely, later replicating regions correlate with B compartments. The genome can be divided into replication domains, which can either be constitutive or developmental. Constitutive domains are constant across cell types, and developmental domains, as their name implies, are those that change during development and are cell type specific. Understanding the mechanisms by which these are regulated is crucial for understanding cell fate transitions. Current experimental approaches like early to late RepliSeq or profiled RepliSeq allow us to measure the genome-wide replication timing profile of cells by BRDU addition, which binds to nascent DNA and can be later immunoprecipitated after cells sorted to finally obtain replication timing as a single signal across the genome, where positive values are going to indicate early replication and negative values will indicate later replication. Previously, we showed that CRISPR-mediated deletion of the joint OCT4-SOX2 nano or OSN binding sites delayed replication timing in a locally early domain in chromosome 16 that contains the DPPA2 and DPPA4 genes, and we call the DPPA domain. The DPPA domain is a developmental domain that is early in embryonic stem cells and is delayed later in differentiation. This prompted us to investigate the effects of OSM binding in replication timing. We obtained cell lines with a Degron system for OCT4, SOX2, and NANOG, and also engineered double Degron cell lines with OCT4 and SOX2 and NANOG, both tagged at once. We were unable to establish a triple Degron or an OCT4 NANOG cell line for unknown reasons. Regardless, this inducible Degron system works by tagging the protein of interest so it can be recognized and degraded upon the addition of the tag. Since we also suspected P300 could be a downstream player in the regulation of the deleted sites in the DPPA domain, we also obtained a P300 commercial inhibitor and analyzed it in parallel to the Degron line. Because we needed a way to compare replication timing shifts across replicates and conditions, we co-opted an in-house differential analysis tool to identify genome-wide replication timing changes that are statistically significant by using a multivariate distance measurement. In addition, and to reduce potential noise, we focused on changes larger than 150 kilobases. We can then classify these changes into delays and advances depending on the direction of the replication timing shifts. We first performed early to late RepliSeq after 24 hours of OCT4 depletion and noted the establishment of a trophoblast-like replication timing program. Since OCT4 degradation has been shown to promote ESC differentiation, we tested an 8-hour DTAC treatment time instead and showed that this allowed us to capture differentiation independent changes. We therefore chose this time point for the remainder of the Degrons with master transcription factors. In the scatter plot, we can see the x-axis represents the replication timing profile of the control, and the y-axis is the Degron or the depletion. So the further a point deviates from the diagonal, the larger the shift in replication timing is. All statistically significant changes are colored depending on what experiment they belong to, and each point represents a 50 kb bin, which is the resolution that RepliSeq was carried out. All of the gray points are not significant changes. In the bar plot, we quantify how many loci, in other words, consecutive bins, are changing, and in which direction, delay versus advance for each condition. And we observed that the knockdown of the pluripotent transcription factors, especially when in combination with each other, causes many more delays than advances. Notably, NANOG depletion causes only a handful of changes, but when depleted alongside SOX2, it causes more delays than SOX2 depletion alone, suggesting cooperativity. Also, P300 inhibition also causes widespread delays. We then wondered how many of these changes overlap with triple OSN peaks, which are those that were deleted originally in the DPPA domain. And this is what we see in figure B. Not surprisingly, most of these changes occur in loci where O, S, and N are expected to be co-bound. And I should mention those that do not usually have either motif binding or ChIP-seq peaks for at least two of the factors, and there are a handful that only have for that of the deleted factor in particular. Likewise, we wanted to quantify how many of these are in constitutive versus developmental domains, which is figure C. So as you can see, most of the changes occur in developmental domains. Then in figure D, we observe an example of one of these delays in developmental domains that occurs in both of the double degrons. This showcases transcription factor collaboration and redundancy. Finally, we wondered how many of the affected regions were common across conditions to better understand the overlap of function of these regulators and replication timing. In particular, we were interested in the double degrons and P300 since these were responsible for the larger number of delays. In the Venn diagrams, we see that advances are not shared across this condition, whereas delays do show an overlap. This means that there's probably an overlap of function greater when it comes to maintaining early replication of developmental domains than there is in those regions that are actually advancing as a consequence of the diagram. In conclusion, we saw that 8-hour Oxford depletion is able to capture differentiation and replication timing changes, and that when we perform diagrams for Oxford, SOX2, and NANOG, and the double diagrams, we see that the depletion of the master transcription factors alone also causes widespread replication timing delays, with the exception of NANOG, which only shows a handful. But then we show that the cooperativity of the master transcription factors, Oxford, SOX2, and NANOG, seems to promote early replication in developmental replication domains, or at least maintain it. And as well, we show that P300 CBP inhibition causes genome-wide delays. While further studies are, of course, necessary to better understand how it is that these transcription factors are maintaining or establishing early replication in developmental domains, these findings are extremely interesting to us, and I will be happy to take any questions in the comments.